Hello everybody, welcome to Biomed Unplugged, and in this video we're going to talk about a paper that was jointly co-authored by Francesco Santarella and myself, Rukmani Sridharan. This paper was selected to be highlighted as a cover image of the journal Advanced Healthcare Materials and is titled Scaffolds Functionalized with Matrix from Induced Pluripotent Fibroblasts for Diabetic Wound Healing. 1 in 10 people live with some form of diabetes worldwide, and 20% of these people develop chronic non-healing wounds in their feet called diabetic foot ulcers or DFUs. Current treatment includes surgical cleaning of the wound and the use of topical creams, but 20% of people with DFUs still end up with a lower limb amputation after prolonged and expensive treatment. One of the main reasons a diabetic wound fails to heal is a dysregulated extracellular matrix or ECM. The ECM is the structural and biochemical soup made of proteins, sugar molecules like glycosaminoglycans or GAGs, and water in which cells are embedded. The ECM in a diabetic foot ulcer often has different ratios of key proteins and GAGs compared to a healthy adult, which has been shown to slow down healing. So our collaborators at Tufts University in the lab of Professor Jonathan Garlick have shown that fibroblasts, which are skin cells, become rejuvenated when they undergo iPS reprogramming. This pivotal technique that won the Nobel Prize in 2012 can convert any mature adult cell into a stem cell that is then capable of becoming any other cell. So bone cells can become nerve cells, for example. The Garlick lab showed that cycling fibroblasts through iPS reprogramming and making them fibroblasts again rejuvenates the cells. They produce more ECM than before, and this ECM resembles the matrix found in young skin that is known to enhance wound healing. Let's call these cells pre- and post-iPS reprogrammed fibroblasts. So, how do we harness the qualities of the post-iPS fibroblast ECM to help heal diabetic foot ulcers? This is where our experience in fabricating scaffolds at the Tissue Engineering Research Group at the Royal College of Surgeons in Ireland comes in. These scaffolds are made of two key components of the extracellular matrix: collagen, the most abundant structural protein in our body, and glycosaminoglycans or GAGs, which are large sugar molecules that form water-trapping networks. Similar collagen GAG scaffolds are the current gold standard solution for treating DFUs using tissue engineering. So we aim to make similar tissue-engineered scaffolds out of pre- and post-iPS fibroblast matrix to see if their composition offered any benefit. We first dramatically increased matrix production from these cells by changing how we cultured them. This matrix was then made into a powder and mixed with a collagen GAG slurry for structural support. This slurry underwent a process called freeze drying that removes all water from the slurry in a controlled manner, leaving behind a beautifully porous, mechanically robust tissue-engineered scaffold. These scaffolds enhance the production of key proteins involved in wound healing like collagen 3, fibronectin, elastin, and some glycosaminoglycans from both healthy adult fibroblasts and, more importantly, from fibroblasts taken from the foot of a patient with a diabetic foot ulcer. Another biological process that is dysregulated in DFU patients is blood vessel formation. A key protein involved in this process is vascular endothelial growth factor or VEGF. We showed that our ECM scaffold supported the formation of new blood vessels and helped both healthy and deceased fibroblasts produce more VEGF. As a final test, we seeded these scaffolds with macrophages, which are key immune cells directing the wound healing response. The ECM-derived scaffolds promoted a robust immune response, allowing the macrophages to dampen or enhance factors involved in inflammation, depending on their chemical cues.
This study has shown for the first time that we can fabricate porous scaffolds with ECM from cells that have been cycled through induced pluripotent stem cell reprogramming. This is exciting as these cells produce an interesting matrix that mimics a younger version of our own tissue. And this is expected to provide an enhanced healing environment. Encouragingly, in this work, that's what our in vitro testing has shown so far, whereby our scaffolds outperform the current standard scaffold. We're now ready to move forward with in vivo testing.
Together, we show that both our ECM scaffolds outperform the current gold standard collagen GAG scaffolds in some of our in vitro tests. The post-iPS matrix led to additional benefits like increased GAG production, increase in VEGF in diseased fibroblasts, and in the production of factors that dampen inflammation. This work was funded by the European Research Council and was initiated by Dr. Cathal Kearney and his team in Royal College of Surgeons in Ireland, and he is now continuing this work at University of Massachusetts, Amherst. We thank everyone involved in this work. This is Rukmini Sridharan at Biomed Unplugged, and we'll see you next time with exciting biomedical research.